If you are black and carry type O blood, the single letter printed on a medical chart holds a genetic signal that reaches farther back than any family surname, farther than the borders of any modern nation, and farther than most written histories ever reach. That letter is not merely a transfusion code. It is a surviving molecular marker whose deepest origins lie inside the African continent in populations that existed long before the main dispersals of modern humans across the rest of the planet. Blood groups themselves were first systematically described in 1901 by the Austrian physician Karl Landsteiner. He observed that mixing blood from different individuals sometimes produced clumping and sometimes did not. From those experiments, he identified 4 principal types: A, B, AB, and O. For the greater part of the 20th century, the system remained a practical tool for safe transfusion and little more. Only when molecular genetics opened the ABO gene itself did the deeper chronological story become visible. The ABO gene sits on the long arm of chromosome 9. It encodes a glycosyltransferase enzyme whose job is to attach specific sugar molecules, the A or B antigens, to the surface of red blood cells. When the enzyme functions fully, the cell displays the corresponding antigen. In type A, most widespread version of that mutation is a single base deletion at nucleotide position 261 in exon 6. The deletion shifts the reading frame, creates a premature stop codon, and produces a truncated, non-functional protein. The red cell surface therefore carries neither A nor B antigen. Genetic surveys of living populations show that this particular deletion, shared by the common alleles designated OZOR1 and O2, is found on every inhabited continent. Because the identical mutation and its surrounding haplotype appear worldwide, the most parsimonious explanation is that the mutation arose once inside Africa, before the principal out-of-Africa expansion that began roughly 60 to 70,000 years ago. Later, independent O mutations have occurred, yet the dominant global form traces to that early African origin. In short, type O is not the ancestral blood type in the absolute sense. Functional A-like alleles are older within the primate lineage, but the common human O allele is nevertheless an ancient African-derived variant that left the continent with the first modern human migrants and has been carried ever since. That single molecular fact already places people of African descent who inherit type O closer, on average, to the geographic and temporal source of the allele than populations whose ancestors left Africa tens of millennia earlier. The same pattern appears when geneticists examine overall human diversity. Africa harbors the greatest nucleotide diversity, the deepest haplotype branches, and the largest long-term effective population sizes of any continental region. Outside Africa, successive founder events progressively reduced variation. Each small group that moved farther from the ancestral homeland carried only a subset of the alleles present in the parent population. The result is a clear geographic decline. Genetic diversity declines with increasing distance from Africa. 2 individuals drawn from different regions inside Africa can differ from each other at more sites across the genome than a typical Northern European differs from a typical East Asian. This is not a cultural claim. It is a direct measurement of sequence variation, accumulated over the longest continuous residence of anatomically modern humans. The same depth is visible on the uniparental markers that pass almost unchanged through successive generations. The non-recombining portion of the Y chromosome, transmitted from father to son, allows reconstruction of a paternal genealogy. In 2013, researchers examining a commercial genealogy sample from an African-American man whose earliest documented paternal ancestor was Albert Perry of South Carolina, identified a Y chromosome that carried the ancestral state at every previously known basal single nucleotide polymorphism. The lineage was designated A00. Subsequent screening located matching chromosomes among the MBO and related communities of Western Cameroon. Age estimates for the split between A00 and all other human Y chromosomes have varied with the mutation rate assumed. The original calculation placed the most recent common ancestor near 338,000 years ago. Later recalibrations using different rate estimates have produced figures in the range of roughly 200,000 to 275,000 years. Even the more conservative numbers still position A00 as the deepest known root of the living human paternal tree, and that root remains geographically concentrated in Central Africa and among the descendants of people taken from that region. A parallel pattern exists on the maternal side. Mitochondrial DNA is inherited almost exclusively from mother to child. The deepest branches of the global mitochondrial tree belong to haplogroup L0, with subclades L0D and L0K reaching their highest frequencies among southern African Khoisan populations. All non-African mitochondrial lineages nest inside the African L3-derived clade. None stands outside the African radiation. Thus, both the paternal and maternal genealogies, when traced to their earliest surviving nodes, point back to Africa. These uniparental signals are reinforced by an independent blood group system, the Duffy antigen. In the great majority of Europeans and East Asians, red cells display either the FIA or FOIB antigen or both. Across large stretches of West and Central Africa, the frequency of the Duffy null allele, FYPBists, exceeds 90%, and in many localities approaches fixation. The null phenotype removes the antigen from the erythrocyte surface. Plasmodium vivax, one of the major human malaria parasites, relies on that antigen as a primary receptor for invasion. In its absence, the parasite's ability to enter the red cell is severely impaired. Although rare, breakthrough infections have been documented in recent years. The historical protective effect remains one of the clearest examples of a geographically restricted genetic adaptation. Additional malaria-related polymorphisms, including the sickle cell allele, certain G6PD deficiency variants, and hemoglobin C, are likewise concentrated in African populations and reflect long-term selective pressure exerted by the disease. Taken together, the ABO-O mutation, the elevated autosomal diversity, the basal Y chromosome lineage A over 0, the deepest mitochondrial branches, and the Duffy null phenotype form a coherent genetic signature. For individuals of African descent who also carry type O blood, that signature is not a distant echo of something that began elsewhere. It is the local continuation of lineages that originated and diversified on the African continent itself. The letter O on a laboratory form is therefore one visible remnant of a far older molecular history, a history written in the genome long before any written record of names or nations existed. The continuity of these markers across oceans is itself a historical fact written into the genome. Between the early 16th and mid-19th centuries, more than 12 million Africans were forcibly embarked on ships bound for the Americas and the Caribbean. Detailed voyage records compiled from shipping logs, port registers, and colonial archives show that the largest single share, nearly 5.7 million people, left from the coasts of West Central Africa, the region stretching from present-day Gabon through the Congo Basin to Angola. Substantial. Additional numbers embarked from the Bight of Benin, the Bight of Biafra, the Gold Coast, and Senegambia. For the mainland colonies that later became the United States, the principal sources were Senegambia and the Angolan coast. These were precisely the geographic zones that still harbor some of the deepest autosomal, Y chromosomal, and mitochondrial variation on the planet. The people taken from those coasts carried with them the O allele that had arisen in Africa tens of thousands of years earlier, the Duffy null variant that had reached near fixation under malaria selection, and the deep-rooting uniparental lineages that had never left the continent. Once in the Americas, the paper records of individual identity were systematically erased. Names were replaced, languages suppressed, and kinship networks broken. Yet the genome continued to transmit the same alleles from parent to child. In the absence of written genealogies, the blood itself became the only unbroken archive. Modern genotyping can therefore recover population affinities that no surviving document records. When large cohorts of African American, Afro-Caribbean, and Afro-Brazilian individuals are compared with reference panels from West and Central Africa, the genetic distances align with the documented embarkation regions far more closely than chance would allow. This recovery of origin does not restore every lost personal history. It does, however, demonstrate that the genetic material that left Africa was not a random or peripheral sample, because the source populations already contained the oldest known branches of the human family tree. The forced migrants carried a substantial fraction of that ancient diversity with them. Subsequent admixture with European and indigenous American populations has layered additional ancestry onto many individuals, yet the African-derived segments retain their chronological depth. Type O blood, when inherited together with those segments, simply makes one component of that depth visible on a routine laboratory report. The same principle applies to the broader pattern of human genetic geography. Outside Africa, the serial founder effects that accompanied successive migrations produced progressive loss of variation. Inside Africa, the long residence time allowed lineages to diverge and accumulate mutations over hundreds of thousands of years. Fossil evidence places anatomically modern humans in North Africa by approximately 300,000 years ago, and genetic clocks calibrated on both mitochondrial and nuclear data place the deepest splits among living African populations in the same broad interval. Out of Africa dispersal that ultimately populated Eurasia, Australia, and the Americas occurred only in the final fraction of that timeline. Consequently, every non-African population is, in genetic terms, a subset of African diversity filtered through bottlenecks. The reverse is never true. African genomes are not subsets of non-African ones. This asymmetry is measurable at multiple scales. Heterozygosity, the average number of differing sites between the two copies of a genome carried by one individual, reaches its global maximum in southern and central African hunter-gatherer groups, and declines steadily with distance from those regions. Private alleles, variants found in only one continental group, are overwhelmingly concentrated in Africa. Linkage disequilibrium, the non-random association of alleles at nearby sites, is lowest in Africa, consistent, with larger long-term population sizes that allowed recombination to break down ancestral haplotypes. All of these quantitative patterns converge on the same conclusion. The main trunk of human genetic variation still resides on the African continent, and the branches that left it are younger and less diverse. When type O blood is viewed against this background, its significance becomes clearer. The allele is not a marker of recent origin. It is a marker that predates the dispersal. Individuals whose recent ancestry lies inside Africa and who also inherit the O phenotype therefore carry both the geographic source and one of the oldest functional variants still segregating in the human population. That combination does not confer superiority of any kind. It does record chronological priority. The mutation that silences the ABO transferase arose in an African population that already possessed deep genetic structure. It then spread both within Africa and outward with the migrants who left. For those whose ancestors remained or returned through the forced migrations of the slave trade, the allele never left its original continental context. The same logic extends to the protective variants that co-occur at high frequency. Duffy null, sickle cell trait, and the common African G6PD alleles are not scattered randomly across the globe. They are concentrated where malaria has exerted the longest and most intense selective pressure. Their joint presence in many individuals of West and Central African descent is the molecular signature of survival under conditions that eliminated large fractions of every generation for thousands of years. When those individuals also carry type O, the laboratory result simply lists one more element of a multi-locus adaptation that was forged in Africa and transmitted intact across the Atlantic. None of these observations requires the invention of new myths. They follow directly from the distribution of alleles, the topology of phylogenetic trees, the archaeology of human fossils, and the archival record of the slave trade. The letter O on a blood type card is therefore neither mystical nor trivial. It is a concrete, measurable remnant of a genetic history that began in Africa, diversified there for the greater part of the human timeline, and continues in the descendants of the people who carried it both within the continent and beyond its shores. The cumulative weight of these independent lines of evidence produces a single coherent picture. The common O allele of the ABO system arose through a loss-of-function mutation on an ancestral A-like background inside Africa, and was already present before the major dispersals that carried modern humans into Eurasia and beyond. The deepest surviving paternal lineage yet identified, AR0, remains geographically anchored in Central Africa and among the living descendants of people taken from that region. The earliest branches of the mitochondrial tree, especially L0 and its subclades, reached their highest frequencies among southern African populations, whose ancestors diverged from the rest of humanity at a time depth measured in the hundreds of thousands of years. Autosomal diversity, private alleles, and low linkage disequilibrium all attain their global maxima inside Africa and decline with distance from it. Protective variants against malaria, including Duffy null, sickle cell trait, and common G6PD alleles, are concentrated in the same continental source. None of these patterns is accidental. Each is the expected outcome of a species that spent the overwhelming majority of its existence inside Africa, and only later sent small founding groups outward. For a person of African descent who also inherits type O blood, the laboratory result therefore coincides with multiple deeper signals. The O phenotype itself is one ancient African-derived allele. When it appears together with elevated African autosomal ancestry, deep-rooting uniparental markers, or malaria resistance alleles, the combination simply records continuity with the source, population rather than derivation from a later branch. This continuity was never interrupted by the forced migrations of the slave trade. The ships that crossed the Atlantic removed individuals, languages, and names, yet they did not remove the genomes those individuals carried. The same alleles that had diversified across West and Central Africa continued to be transmitted in the Americas, generation after generation, until modern sequencing made them readable again. It is important to state what this evidence does not claim. It does not rank blood types or populations by intrinsic worth. It does not assert that every person of African descent carries every deep lineage, nor that type O is unique to Africa. The O allele left Africa with the early migrants and is now nearly universal among indigenous populations of the Americas and common in many other groups. What the data do establish is chronological and geographic priority. The mutation, the diversity, the basal Y chromosome, the basal mitochondrial branches, and the malaria adaptations all originated and first reached high frequency inside Africa. Populations outside Africa carry subsets of that variation filtered through successive bottlenecks. Populations whose recent ancestry remains predominantly African retain larger fractions of the original depth. This chronological priority has been obscured for centuries by historical narratives that treated Africa as a brief prologue rather than the long main body of the human story. Written records, colonial archives, and popular accounts often began the human timeline after the out-of-Africa dispersal, thereby rendering the preceding hundreds of thousands of years invisible. Genetic data reverse that inversion. Fossils of anatomically modern humans appear in Africa by approximately 300,000 years ago. Genetic estimates for the deepest splits among living African lineages fall in the same broad interval. The dispersals that populated the rest of the world occupy only the final fraction of that span. Consequently, the statement that African genomes contain the root of human variation is not a metaphor. It is a direct description of measured sequence diversity and phylogenetic topology. When the letter O appears on a blood type report for someone whose ancestry traces to those root populations, the report is registering one visible fragment of that deeper archive. The fragment is small. A single locus cannot reconstruct an entire genealogy. Yet it is not random. It is an allele whose origin, age, and geographic distribution align with the larger pattern established by the rest of the genome. In that limited but precise sense, the blood type points back to the same continental source that every other major genetic system also identifies. The practical consequence is straightforward. For individuals and families whose documentary histories were deliberately severed, the genome supplies an independent record that no bill of sale or plantation ledger could erase. Type O blood does not by itself name a specific village or ethnic group. Combined with denser, autosomal and uniparental data, it can, however, narrow the range of possible source regions to the same West and Central African coasts from which the majority of captives were taken. That narrowing restores a measure of geographic and temporal context that paper records alone cannot provide. The same restoration applies at the scale of the species. Once the full depth of African genetic variation is acknowledged, the familiar narrative in which humanity began only after leaving Africa becomes untenable. Humanity began diversified and spent most of its existence inside Africa. Everything outside is a comparatively recent and less diverse extension. People of African descent who carry type O blood therefore stand in the measurable layers of the genome nearer the trunk than the outer branches. That position is a matter of time and place, not of rank. It simply corrects the historical amnesia that once treated the trunk as a footnote. Science continues to refine the numbers. Mutation rate calibrations shift age estimates by tens of thousands of years. New ancient genomes add intermediate nodes to the trees. Breakthrough infections show that even Duffy null is not an absolute barrier. None of these refinements overturns the central geographic pattern. Africa remains the source of the deepest lineages, the highest diversity, and the earliest functional variants still segregating in living people. Type O blood, when inherited in that context, is one more datum consistent with the pattern. The next time the letter O appears on a hospital form or a blood donor card, it can be read for what the molecular evidence shows it to be: a surviving fragment of an African origin allele that predates the peopling of the other continents. It does not speak for every ancestor or every chapter of a family's history. It does, however, speak for a continuity that written history once tried to erase and that the genome never forgot. That continuity is the true bloodline the data reveal. Not a claim of exclusivity, but a record of chronological depth rooted in the continent where our species first became human and where the greater part of its genetic variation still resides. If this account of the genetic evidence has clarified something about the age and origin of the blood you carry, share it with someone whose family history was also interrupted by the Middle Passage. Leave a comment describing what your own ancestry testing or family records have shown. Subscribe if you want further examinations of the DNA evidence that continues to rewrite the timelines we once took for granted. The blood remembers what the documents erased, and the tools to read that memory are now in our hands.